Hello and welcome to this joint webinar, Keep Your Flow, presented by ibidi and us at Face Holographic Imaging, featuring Markus Bosteen from Novo Nordisk. Markus will show his microfluidic assay setup and discusses how he uses non-invasive holographic microscopy together with impedance measurements and the ibidi cell culture and the flow products to investigate how pathological flow patterns contribute to the progression of atherosclerosis. Did I pronounce that right?
Perfectly.
Yeah. My name is Lisa Bodily. I'm the communication manager here at Face Holographic Imaging and also part of the PHI scientist team. And I will be your host for today. And with me here, I have Peggy Benisch, head of sales at ibidi. Hi, Peggy.
Hello,
great
everyone.
to have you. Thanks. After Markus's talk, before we go into the Q&A session, Peggy will briefly share some key information about using the ibidi flow system and the impedance measurement system for successfully setting up microfluidic assays. And I will also give you a quick in a nutshell summary on the HoloMonitor live cell analysis system, technology and applications for cell research. Lastly, before we jump into the presentation, I would like to inform you that this webinar is recorded and you will receive the recording after the live event. And also for you listeners, you have a control panel where there is a question box. So please feel free to use that one anytime during the presentation to ask any of the speakers, especially Markus, your questions. And we will have time at the end of the webinar to address those questions. If we can't manage all of them, no problem. We will try to get back to you as soon as possible via email. And yeah, I guess with no further ado, I will introduce Markus a little bit more. Markus studied molecular biomedicine and graduated from the University of Copenhagen in 2011. First got interested in the topic of vascular research during his bachelor studies and continued that throughout the master's and the PhD with a focus on endothelial function during inflammation, angiogenesis, and the mouse model of atherosclerosis. And after his PhD, Markus worked extensively with cell-based assays and immunoassay development, as well as validation as a scientist and research scientist in industry. And since 2021, he has been working as a senior scientist at the Vascular Biology Department at Novo Nordisk in Denmark. So welcome, Markus. And I will...
Thanks a lot, Lisa.
I will let you have the screen.
I'm gonna jump right into it then, since we don't have too much time and a lot to go over. So as Lisa said, I work in the Department of Vascular Biology at Novo Nordisk, and in my department we have a strong interest in atherosclerotic cardiovascular disease, and we also have a very strong desire to come up with new treatment options for this disease. That means treatment options that really goes beyond the traditional blood pressure lowering and lipid lowering therapies that already exists. And the reason for this is that in spite of these very effective treatments, then atherosclerotic cardiovascular disease, it continues to be the #1 killer within CVD, and it's really also the #1 killer compared to any disease in the world, unless you count all cancers as being one disease. So because of this mission that we have to come up with new treatments, we recently turned our attention to blood flow. And then you might ask, so why is blood flow so interesting? And it's really because blood flow and blood flow patterns, they are strongly correlated with sites of atherosclerosis development. And that's actually also what you see in the first slide here. So this is a guy. You can tell by the ear up here, the left ear, and some beard next to it. And he's had his neck cut open to expose the carotid artery. And that's the one you can see up here. And you can also see that he has signs of atherosclerotic plaque development at the point where the carotid artery, it bifurcates into the internal and the external carotid artery. So the question is really, what's so special about this exact anatomical location? One thing that is special is the flow patterns in this area. So in the middle here, you see the blood flow speed in the same exact anatomical location. And you should be aware that the red color indicates high flow speed, and the blue color indicates a low flow speed. And to the right, you can see again the same anatomical location that has been color coded according to the time average blood flow that is measured in shear stress as dynes per centimeter squared. And you can even see that the shear pattern or the shear profile, which is measured over one cardiac cycle up here, it's very different between the area of low shear stress, the blue area, and the area of higher shear stress, the green area immediately above. And also you see that the shear stress profile below, it even indicates that the flow direction is sometimes reversed, as you can see by the negative values on the y-axis. So question is, why is this then important? I mean, why do we even care? And as I just told you, then the anatomical sites of low and bidirectional shear stress, it correlates with the sites of atherosclerosis development. And to really make a very long story short, then the endothelial cells, which are the cells that line the inside of blood vessels, they respond to shear stress. And endothelial cells also play a major role in initiation and also in progression of atherosclerosis. So on the next slide, very simplistically put, then endothelial cells, they respond to flow. And if the flow is normal or healthy, that means if the flow is undisturbed, it's laminar and it's unidirectional, then the endothelium can function normally and preserve a healthy artery. On the other hand, if the flow is low or turbulent or bidirectional, then the endothelium becomes dysfunctional. And this, together with other risk factors, will promote development of atherosclerosis and in the end cause a fatal occlusion of the blood flow to potential critical tissues. So just to convince you that the endothelial cells actually do respond to flow, I brought a short time-lapse video that's showing human coronary artery endothelial cells responding to flow. And this video has of course been captured using a holographic microscope from Phi. So what you see here is a culture of cells that is running for 67 hours in total. And you can see that over time, the cells start to become more morphologically homogeneous, and they also start to align with the direction of flow. And you should keep in mind that in this image, the cells are pseudo-colored according to height, or what is really the optical thickness, which is also the profile that you see in the graph in the bottom right corner. So you see over time that these cells really nicely align with the direction of flow. I hope this really convinces you that endothelial cells, they actually do respond to flow. And I will just briefly explain in the next slide what types of mechanisms these cells use to sense the flow and the shear force that are generated by fluid flow. So endothelial cells, they have developed multiple mechanisms to sense flow. And this slide just summarizes a few of them. And in the interest of time, I'm not going to go through all of them in detail, but I'm just going to leave it by saying that various structures, including cell-cell junctions, membrane structures such as caveolae and also primary cilium, they all participate in shear sensing by transferring the shear force into and also onto the cells. And even the glycocalyx is very important in this context, and shear force can also be sensed and transmitted by single molecular structures, including ion channels and tyrosine kinase receptors and also G-protein coupled receptors. So all these shear sensing mechanisms, they signal inside the cell, and they can influence basically every imaginable function of endothelial cells, and this includes blood pressure regulation, it includes inflammation, angiogenesis, and finally, and also very importantly, it also influences thrombogenesis. So finally, I'm going to go into the main point of this webinar, which is how to set up microfluidic assays to investigate endothelial response to flow in vitro. So in my lab, we use the perfusion pump system from ibidi, which is the one that you see in this slide. And the way that it works is basically by pumping culture medium back and forth between two syringe reservoirs. And the system then has a really clever valve system, which is controlled by a standard laptop computer. And these valves, they continuously regulate the flow so that it's always either unidirectional, bidirectional, or pulsatile, depending on what you want it to do. And at the same time, the culture medium is then pumped across a microslide that holds the cells. And that's the one you can see in the right illustration. And this is really how the cells are exposed to the fluid flow. This type of setup, it also allows for a highly customized experiments because you can connect these microslides in almost any way you want. So you can either manipulate the flow speed, you can manipulate the flow pattern, and you can also manipulate the number of replicate slides if you want to do that also. And what is really neat about it, and also what we do a lot in my lab, is then you can then pre-treat the cells before you connect them to the flow system. And that way you can further increase the number of conditions that you can include in the same experiment. And then what you can also do is then you can connect this pump system serially to an electrical cell substrate impedance sensing platform. And this gives you a readout of endothelial barrier function at the same time. And that's what you can see sitting here on the top shelf of the incubator. And what you can then do is you can connect the pump system to the holographic microscope from Phi, which will also give you live cell imaging at the same time. And that's what you see over here in this image, where I've connected a 6-channel slide to increase the number of replicates and experimental conditions even further. So by using this setup, you can obtain not only the mRNA expression profile by simply harvesting the cells at the end of the experiment, but you can also get a real-time measure of barrier function and migration, proliferation, motility, and morphology, all from the same experiment. And then you can even harvest the culture medium either during the experiment or at the end to look at the secretory profile of the cells. And if the cells are not used for RNA isolation, they can also be used for staining purposes or other techniques, including Western blotting and other immunoassay methodologies. So this slide is really just to give you an impression of the type of imaging that you get from the holographic microscope. What is really cool about the technology is that you can actually create 3D projections of your cells, just like the image that you see here. And again, this is of course endothelial cells that you see in the picture. And you can also see from the blue line on the image that you can get a high profile of the cells, and that makes it possible to calculate an optical volume if you also want to do that. And again, I should just remind you that the cells are pseudo-colored at this time according to their optical thickness. That's how you get these different color gradients over the cells. So in this slide, it's really meant to just show you how morphology data is processed and also quantified by the software that controls the holographic microscope. So what you see here are 3 different conditions. You have the unidirectional flow, you have the bidirectional flow, and you have the static condition. And you can clearly see that the cells, they align with the directional flow. And in this case, the direction is left to right for the unidirectional condition, and then alternating left to right or right to left for the bidirectional flow. And what the software does is then to automatically find the edges of the cells and then calculate a lot of different morphology parameters. And I've just plotted 2 of these parameters in the graphs so you can see how the cells they change over time. And even though the actual effect size in terms of cell elongation is actually relatively modest, it really doesn't change very much numerically, but then the software is still able to pick it up as a highly significant difference. And personally, I really like this graph a lot because it not only illustrates endothelial response to flow, but it also just shows how much data is actually handled by the software. So for instance, in this graph, the sum of data, or the sum of the data points is actually 1.5 million that's visualized in a single graph. And the software for the holographic microscope takes care of all of this for you. So here's just another example of the type of data that you can get using real-time holography. And this time I've made the software count the number of cells over time and then plotted it. And again, you see that there's a clear difference between the different types of flow. And you can also see that from the holographic images to the right. And you can even see an effect of TNF-alpha treatment that was given after 67 hours of culture. And in this experiment, I also harvested RNA for qPCR analysis to try and correlate some of these observations on the microscope with a more mechanistic readout at the transcriptional level. And what I found is that there's a really good correlation actually between the expression levels of 2 very important cell cycle regulators, including cyclin D and E2F. And the observations that I made on the microscope even correlates with other cell cycle regulators such as P21 and also P27 that I'm not showing here just due to the lack of space on the slide. So here's again another example of motility data from the same exact experiment that you just saw on the previous slide. And again, the software automatically generates this by tracking each individual cell over time, and then a cell speed is calculated and expressed in micrometers per hour. Once again, you can see that the cells, they respond differently depending on the type of flow that they are exposed to. And you can also clearly see the effect of TNF-alpha treatment after 67 hours, which in this case causes the cell motility to go up. So in the next couple of slides, I want to show you a couple of examples how electrical cell substrate impedance sensing can be used to gain some additional information on cell behavior under flow and in real time. For this setup, you need the ESIS platform that you see here in the incubator sitting next to the ibidi flow system. And you also need special micro slides with the gold electrodes on the bottom. And that's the type of slides that you see in this picture. So what I did in this experiment was to first apply a unidirectional flow on both of the fluidic units of the flow system. So that's the same flow going on 2 different microslides. And then after 1 day, the flow was changed to oscillating flow on one slide and unidirectional flow on the other slide. And by the way, in this case, oscillating flow has the same meaning as bidirectional flow. And what I did after 1 day is then introduce electrical wounding on both of the slides to denudate the electrodes of cells. And this causes the electrode capacitance to rise to all the way up to a maximum value of about 3.5 nanofarads. And then what happens over time is that the cells will then start to migrate back onto the electrode. And this can be measured as a decrease in capacitance over time. And once again, it's pretty clear that the cells, they actually respond differently depending on which type of flow they have been conditioned to prior to the electroporation. So in this case, cells that have been exposed to oscillating flow, they migrate faster compared to cells that were conditioned by the unidirectional flow. And this response is also very much in line with what you just saw on the previous slide, where the cells were less motile under unidirectional flow compared to the bidirectional flow. So this is another experiment that was run for 92 hours in total, using a special bifurcated slide that can generate gradients of different flow patterns across the same microslide. And again, you clearly see morphological differences when comparing the oscillating flow and the unidirectional flow. And because the slide is bifurcated, then you also see the cell alignment on a unidirectional flow, that it actually follows the direction of the slide, and therefore also the direction of flow. So in this case, the cells were exposed for 2 days to either unidirectional or oscillative flow, and then they were wounded using electroporation, just as in the previous experiment. And this time, the cells were monitored for an additional 2 days after wounding. So first of all, this allows you to follow their migration, just as in the previous experiment. And again, this was much slower on the unidirectional flow compared to oscillating flow. But it also allows you to follow the barrier function over time. And as you can see, it's pretty clear that the barrier integrity is better on the unidirectional flow. So these are the traces up here, when you compare it to the bidirectional flow, which are the traces below. So this is exactly the same experiment as in the previous slide. And it just shows you that even within the same bifurcated microscope or bifurcated microslide, you can actually observe morphological differences depending on the exact location in the slide, and therefore also depending on the exact flow pattern at that location. In this case, the flow pattern, it differs exactly because I'm using the bifurcated slide. So what you can see that over time, the effect of flow differences can be measured as a difference in barrier function, so that the cells in the location of the lower shear stress, which is the green area, or what corresponds to the green area in this flow profile that you see up here in the corner, then they form a less tight barrier compared to the cells that are located in the area of high shear stress. So that's the green line up here. And again, this is exactly the same experiment as in the 2 previous slides. And this time I'm just zooming in on the individual traces that are captured by the ACES platform. So this is really meant to show you that you can get another readout from the same data. In this case, it has to do with cell motility, or what is called cell micromotion. And clearly you can see that it's not only the trajectories of the traces that are different, so indicating that barrier function is actually declining over time for cells that are exposed to bidirectional flow, but it's also the pattern within the traces that are different. So showing that cells that are exposed to bidirectional flow, they actually display more micromotions when they are compared to the unidirectional flow. And once again, I tried to correlate this with a mechanistic readout at the expressional level. And it has been shown previously that micromotion, cellular micromotion, is associated with a low MEC5 and ERK5 signaling. And this is actually the pathway that ultimately ends up in upregulation of transcription factors called KLF2 and KLF4. And when I look at the transcriptional level, I do actually see that these factors are downregulated on the bidirectional flow, which is consistent with this lower MEC5 and ERK5 signaling. So again, even at the transcriptional level, there's a correlation between what you observe in the ACES system or the holographic monitor and what you also see at the transcriptional level. So this is really my final slide, or the final data slide, and it's meant to demonstrate that a lot of proteins that are important for endothelial function, they are regulated by flow at the expressional level. So if you see, for example, endothelin-1 and angiopoietin-2, and these are both factors that are involved in endothelial dysfunction, they are also upregulated by bidirectional flow or disturbed flow. And also IL-8 and CCL2, also known as macrophage chemotactic protein 1, they are heavily regulated by the combination of both bidirectional flow and TNF-alpha stimulation. And also NOS3, which is also known as eNOS, and even NOx4, they are practically inversely regulated by flow and TNF-alpha stimulation. And then finally, you see that the heme oxygenase 1, which is an enzyme that's really important for dampening the inflammatory response in endothelial cells, that's also upregulated by the unidirectional or healthy flow, you could say. So just to conclude, then I hope that by showing you this data, I've convinced you that the endothelial response to flow is really important for health and for disease. And I also hope that you find it convincing that there are really many aspects of endothelial cell biology that are affected by flow and by different flow patterns. And then finally, I hope that I've shown you that the ibidi flow system in combination with the ACEs and holographic imaging is really well suited for investigating endothelial response to flow. So with that, I would just like to thank you for listening, and I'd be happy to take your questions.
All right. Thank you, Markus, for letting us in to your assay setup and showing us how you maximize the amount of data from every single flow experiment. I had a short connection issue on my end, but I hope you guys were okay.
We were probably fine. I just got a bit nervous. Where's Lisa? But you're back.
Yeah, yeah, I'm back, but it's great. The show must go on, right? So listeners, please, if you have a question to Markus in his presentation, use the control panel. I've already seen one coming in, but one is only one. We want more questions for Markus. We will address them after in the Q&A. But before we get there, as promised, first Peggy and then second I will give you short the key takeaways about the equipment that Markus have used in his cell culture and the flow setup. And I think, Peggy, how about you start?
Sure.
I will have a look. Giving you the screen, 1 second.
Okay.
There you go.
All right. So I hope everyone can see my screen. Actually, I don't have to add anything because Markus was such a great ambassador for both ibidi and PHI. So thank you for that. Still, I would like to give it a try to summarize all the tools Markus used for his experiments just as an overview. And of course, the most important part is the pump system. You need to have cells under flow somehow. And what Markus performed was once a non-visual analysis, so impedance measurement, and a visual analysis to analyze the cells under flow via microscopy. Some words about the pump system itself. The ibidi pump system is an air pressure pump, so it generates air pressure to move the liquid forward. And as Markus also very nicely showed, it's a closed system. So we have a special mechanism to make sure that the liquid inside of the slide is passing the cells again and again. And this feature, together with the possibility to place part of the pump system in a regular cell culture incubator, you can do long-term experiments for hours, days, or even weeks. And different flow types as well, like unidirectional or pulsatile or oscillatory. But what I also would like to talk a bit more in detail are the holders for the cells, so the labware. With our pump, you can easily connect any slide that comes with a female lure port, because we use the male-female lure connection system, which is quite convenient. And for each application, so impedance and microscopy, you would need specialized labware or tools. So for the impedance measurement, a slide would be convenient with gold electrodes, as you can see here for the ESIS impedance measurement system. Let's have a closer look at that. Markus already showed you how it looks like. Here's some more details on that. This is the slide. Here's the channel, and the channel is connected to the pump system. You can see here the tubes, and this is part of our pump system. And inside of this channel are the tiny gold electrodes. And what the impedance system is measuring is the cells that are sitting on this gold electrode, because the gold electrode actually transmits the current. And what's beautiful on those two pieces of two different instruments is that you can place both in an incubator. And I think everyone knows that in the incubator with the right settings, the cells are happiest. And this is how it's possible to do experiments for several hours or even days. Let's move from the impedance to the microscopy. What's important for microscopy? First, to have labware and holders for cells with perfect optics, so good optical properties. Here I show some relatively simple slides, but we have a whole range of different slides. And what all of them have in common is that they come with a cover slip bottom, so either in glass or in polymer even tissue culture treated. And since it's a cover slip, imaging, they're perfectly suitable for imaging and microscopy. And we have different designs. So we have relatively simple straight channels for shear stress experiments. So when you need defined shear stresses and need to calculate them, defined geometry is quite important. So straight channels, it's easy to have the correct shear stresses in those. Same goes for the Y-shaped slide with the bifurcations. But on the other hand, we also offer solutions for anyone who wants to have cells in a gel perfused or would like to generate spheroids or organoids and cultivate them for several days. This one would be of interest for you. This is just a quick introduction to show you how versatile our slides can be. And if you're interested in any of those, go to our website and request a free sample. So those are the probe holders or the holders for the cells with the good optics. And what else do you need? For microscopy, you need a microscope. And with this, I would like to hand over to Lisa from PHI to introduce you to the HoloMonitor in more detail. Okay, that was all from my side.
It's great, Peggy. It's a very smooth handover because isn't ibidi always about happy cells? Right?
Yes, we have.
Yeah. No, we are also. That's also what we're saying all the time. Like happy cells. It's what we need. Let me just pull up my presentation for you guys as well. So, just 1 second. There we have it. I just saw like so many questions coming in, so just had a look at that. So before we get there, I would take like 3 minutes just to show you guys a little bit about the HoloMonitor system. That is a live cell analysis system, and yeah, Markus, you have basically said it all already. But what I thought I just summarize it as well. For those of you who haven't seen it, it's a live cell system based on digital holography that has been used actually in over 200 different peer-reviewed publications worldwide so far. And the HoloMonitor software records images and quantifies therefore many different aspects of the daily life of cells, as you said, Markus, from proliferation, movement to morphology. And it does that without any stains or labels. So this is a label-free, non-invasive method. And the results come directly from the incubator. So there we go, Peggy, happy cells in the incubator. And usually to emphasize this a bit more, I usually show how digital holography works. So maybe that answers one or other questions you might you have. How did Markus get that data? So HoloMonitor uses, as I said, label-free digital holography, and we have a low power laser beam that goes into the back of the system and is split into two. One is the reference beam and one is the beam that goes through our cell sample. And when it goes through the cells, it doesn't affect them in any way. However, what does happen is that when light passes through the cells, is that the light gets shifted. It's called a phase shift, which we can detect. And when reference beam and sample beam meet again, a hologram is created, as you can see on my slide here. And from that hologram, the computer reconstructs the cell image digitally. So everything you've seen, that's a digital cell image. And this pattern, that hologram, we see now on the left-hand side, that is also our raw data. which is used to quantify that light phase shift. And that in turn is used to calculate, for example, every thickness part of the cell in every single pixel of the image. So that's a lot of data. The software can then recreate a cell image based on that thickness calculations and can give you pictures in 2D or even 3D. And as Markus also said, beautiful colors, they're all software generated. It's a label-free method. Here we have the blue as a background and sort of white, turquoise, grayish cells showing different thicknesses of the cells. And yes, once we have the images taken, every analysis begins with cell identification. And as every holographic image contains the data about the thickness of the cell, the software can determine what is cell and what is background. And it determines that by identifying the cell boundaries with a very high accuracy for cells in every different shape and size. And of course, once we have that, then we can pull the data. So with this slide, I just want to show you the different types of data you can pull, either from a single cell, over 30 different morphological parameters, or from a single image, you can get the single cell data or the population data. Or if you take multiple images from the same cell spot, then you get time-lapse movies. For example, like I think that's a cell dividing here right now. And from time-lapse movies, we can then see how the cells have moved, track them, how fast were they, where were they going, how have they changed the morphology. Like, for example, in this example, the cell volume dropped due to cell division. And we can also have a look at different cell fate in the cell family tree plot we are providing, and you can have a look at different cell cycle lengths. So there's a lot of data in there. So yeah, one key advantage with the HoloMonitor system that it's all data is stored in the images of the cells. So you can reuse your imaging results to get basically multiple results, but from the same cell sample. I've just put a couple here, and for example, we always develop these assays with the researchers and our partners in mind, and actually with ibidi together, we have developed our wound healing assay, which is completely, like 100% compatible with the ibidi products, just to make it easier for you guys. And that is a powerful tool, because then you will save time hands-on, not having to set up all over new assays to get multiple types of data. So yeah, it's time and cell consuming, I guess, or not time and cell consuming. I promise to keep it short, so if you want anything more, please visit us online, get in touch with us if you have any questions or would like to have a demo. And speaking of questions, let's go to the Q&A. So, Peggy, Markus, do you have your microphones on? Yes, you're all ready.
Yes.
So let me have a look. So everyone, if you still have questions, please use the question box. And Markus, I've got something for you. How do you stimulate your cells while they are under flow?
Yeah, so that's a really relevant question, because of course you might think that you need to stop the experiment in order to treat the cells or somehow interrupt the flow, but actually that's not necessary, because ibidi also can provide you with these special injector ports that you can connect to the tubing serially. So these injector ports, they have a small membrane where you can insert a syringe, and you can inject your stimulant directly into the flow system while it's running. So that's a major advantage, because you don't interrupt the flow or the experiment in any way.
Mm-hmm. I guess a follow-up question here, like how long do you usually culture your cells under flow?
Yeah, so normally what we do on a routine basis is that we keep the flow going, or the entire experiment going, for about 48 hours. But We also go up to 3 days, and the longest one that we've done is 4 days. But as Peggy also explained, it is actually possible to go up to even weeks just by changing the reservoirs to a larger volume or larger syringe volume, or you can even exchange the medium underway. But of course, doing a medium exchange is a little bit more of a hassle. But if you use the large volume reservoirs, then you can run uninterrupted for, yeah, I'd say at least 2 weeks. The only limitation is that you get if you have a lot of tubing and a lot of equipment connected in series, like I do, then you get some evaporation from the silicone tubing. So that should be kept in mind.
Good point, Markus. Yeah.
I've
Nothing
got a
to add
question here. Another question for Markus from Carol. Can you distinguish between two cell types using fluorescence?
That's a good point. I mean, in this case, I'm not using any type of fluorescent markers, but of course you could do that at the end of the experiment. So simply fix the cells and do a staining afterwards. But using the holographic microscope as it is now, the only way to distinguish is by morphology. Yeah. Otherwise, you have to do it at the end of the experiment. But that can also be done. You can do staining.
Yeah, I guess while we're on the topic, we've received 2 more questions about fluorescence here. Yeah, what are you planning next, Markus? I think that question goes to you.
Yeah, I think that's a very relevant question. So as you may be aware of, then PHI, they are working on incorporating fluorescence into the holographic microscope. And fortunately, we have exclusive access to the demo version of this instrument. So that's some of the experiments that I'm working on now to actually combine holographic imaging with fluorescence imaging. And that opens up a whole range of new possibilities, like live cell imaging with the fluorescence, so you can even distinguish the different cell types, or you can even use different fluorescence markers, such as dyes that are sensitive to reactive oxygen species, or apoptotic dyes, or I mean anything you can think of really. So that's on the to-do list, and then hopefully can be included in the next webinar.
Yeah, we should definitely schedule another one then.
Absolutely.
Speaking about different microscopy methods, that's a question for Peggy, I think. Can you use the slides, I guess the micro slides, also for confocal microscopy?
Oh yeah, that's definitely also possible. So all kinds of light microscopes, not just the holographic ones. Yeah.
Yeah.
Because it's a cover slip bottom 1.5, so it's the standard. Right.
Yeah. So if there's any more sort of follow-up questions on that, we can probably answer that by email as well, a bit more extensively,
of course.
right? Exactly.
Happy to answer any questions, so bring them in.
Let me scroll through here. Markus, this is for you. How do you calculate the level of shear stress in your customized flow setup?
Yeah, so that can be a little bit more less straightforward than normal, because if you use the standard setup provided by ibidi, then the software actually will almost calculate this completely automatically, depending on which type of perfusion set you're using, which is color coded in advance. So you can basically just plug that information into the program. But when you're using a custom setup, then some of the settings will not be valid anymore. So you have to do a manual calculation. So it's really about measuring the flow rate and then using so manually measuring the flow rate and then using some standard values for the slides that are also supplied with the slides. So there's a little bit of a manual calculation in there. but it's actually normally part of the standard QC that you would do anyway if you do it the right way, even though you have the values up front. So it's not a lot of extra work.
Sounds good. Let's see, what else do we have here? For long-term experiments, how do you assure a CO2 equilibrium of your medium?
Yeah, so I mean, because the tubing is actually gas permeable, and because you have the perfusion going on all the time, then that is actually sufficient to ensure a proper gas exchange. And I think that, and please correct me if I'm wrong, Peggy, but I think that even the microslides are also somewhat gas permeable. So
exactly,
yeah,
they
so
also
as long as everything, so as long as everything is in the incubator, then it will be equilibrated automatically, simply by convection.
Anything more to add, Peggy?
No, that's fine. I mean, on the pump system, there are also filters, so gas exchange also happens there, so perfectly fine.
Let me just see. I got a question if the HoloMonitor is limited to 2D cell culture, or if we can also work with 3D systems. That is a very good question. It's the holographic microscopy is designed for, well, most people use it for monolayer adherent cell cultures, but as we see, we have also flow setups, but we have also customers using 3D cell cultures, for example, with Matthew gels. But I think I can just forward you after the webinar a couple of publications on that topic. And if any one of you guys is using a more 3D system, just reach out to us and our application specialists can sort that out with you. Let's see, what else do we have here? This one we had. All right, what types of cells we can image? Yeah, I kind of said that earlier. Like many customers use different cell lines, cancer cell lines, especially stem cells and cell lines, as it's a non-invasive technology. Primary cells, maybe here also just a referral back to our application specialist who you can just contact with your cell type, and then we can have a look who else might have worked with it. We have also the publication summary on our website, which you can search for your cell type as well, just as a hint here. All right, actually looking at the time, there's a couple more which just popped in, but I think we take them later by email, so we appreciate everyone's time here for today. Markus, thank you so much for your informative presentation. Thank you so much.
Thanks for having me. It was a pleasure.
And for our audience, please don't forget to fill out the survey after the webinar. That would help us immensely to become better at what we're doing. We really appreciate your feedback. There's also 3 free handouts you can take if you look in your control panel, so you're free to take that. And if you have anything, please reach out to us anytime, Markus, Peggy, or myself. And I think the only thing left to say is thank you for attending today and have a great day. I guess bye, everyone.
Bye, everyone.
Bye.
Bye.
Thanks for listening.
Thanks for listening in. Exactly. Ciao.